Hi everyone, this is Yue Zhao. Welcome to the class Introduction to Bioinformatics. I will teach Module 3, Sequencing Data Analysis. In the following 4 weeks, I will give 4 lectures and 4 hands-on lab. You can read the overview section of this module to get a better idea what will be covered in the following 4 weeks. I have also posted some reading materials for this lecture and the next lecture. And after Friday's lecture, I will post the homework for this week. Before I start today's lecture, I want to thank Professor Li Juncheng for providing teaching slides. Okay, let's start with the so-called most fundamental operation in bioinformatics, pairwise sequence alignment. In this lecture, we will talk about alignment basics. Why are we comparing biological sequences? Why are we comparing biological sequences? Here, the biological sequence include the DNA sequence and the protein sequence. First of all, there is a paradigm in biology that a similar sequence may have similar structures and may lead to similar functions. So in this case, the similarity between sequences can help us infer the possible function of unknown new sequence based on the function of known sequences. Secondly, from an evolutionary point of view, similar sequences in different species often mean that they have a common ancestor, so-called homology. In fact, the similarity between sequences is one of the foundations for constructing evolutionary trees in phylogenetic study. There are also many practical bioinformatics applications. For example, similarity searching of database for protein structure prediction and annotation, or assembly of sequence reads into a longer construct, such as a genome sequence, and mapping sequencing reads to a known genome or transcriptome. For example, resequencing to look for SNPs, indels, and large structure variants, mapping transcription factor binding sites, or for transcriptome study, looking for fusion genes or splicing sites. So pretty much all next-generation sequencing data analysis use the sequence alignment, and we can say that pairwise sequence alignment is the most fundamental operation of bioinformatics. So next, let's look at the homologous detection. Sequence comparison is most informative when it detects homologs. Homologs are sequences that have common origins. For example, they share a common ancestor. They may or may not have common activity. Homologous sequences may be either orthologs or paralogs, depending on if they have common activity or not. So how to define if the two sequences are homologs? There is 2 matrices, identity and similarity. Actually, any pairs of sequences may share a certain level of identity and similarity. So if these metrics are above a certain level, we often infer homology. Orthologs are homologs that have common activity. They are produced by speciation that have diverged due to divergence of the organisms they are associated with. Also in Greek means straight. It implies direct descent. For example, here, the bird and the snake, they share the common ancestor dinosaur, and the sequence in dinosaur has changed over time, especially the change is happened at the time point the speciation event happened. On the other hand, paralogs are homologs that have different activities. They are produced by gene duplication. They represent genes derived from a common ancestral gene that duplicated within an organism, and then subsequently diverged by accumulated mutation. Para in Greek means alongside of. For example, in this single species, the original sequence got diverged, got duplicated, and during this duplication process, there are certain mutations, and also it got further diverged due to many mutations along the way. In practice, determining orthologs versus paralogs can be a very complex problem. Sometimes genes get lost after duplication. It's really hard to trace back. And most of the time, we lack of the knowledge of evolutionary history. And sometimes there is a weak similarity because of evolutionary distance. And also, homology does not necessarily imply exact same function. So it may have a similar function at a very crude level, but play a different physiological role. The sequence changes during evolution. There are three major types of sequence change. One is mutation or substitution. Second is deletion. The third is insertion. We still use the dinosaur bird and the snake as the example. The first one is the dinosaur sequence. CT becomes CA. There is a mutation happen or substitution happen. T is replaced by A. And this mutation is very likely happened before the speciation. Then next, after the mutation, the G got deleted. So G is disappeared. And then there is an insertion happen between these two T's. So there is the alignment view. It's a great tool to visualize the sequence similarity and the evolutionary changes in homologous sequences. So here, we align sequence A in the first row, sequence B in the second row, and then we compare these two one by one base by one base. Then mismatch is represent the mutations and the substitutions and is represented by the column here. And the gaps represent the insertion and the deletions. And the small, the short line represent match. Here in this example, there are 5 match, 1 mismatch, and 2 gaps. Unfortunately, finding the correct alignment is difficult if we do not know the evolutionary history of the two sequences. So there are many possible alignments. In this case, there are 3 possible alignments, and which alignment is the best? One way to judge alignments is to compare their number of matches, insertion, deletions, and mutations. The first alignment here has 4 matches, 3 mismatches. The second one has 6 matches and 2 gaps. The third one has 5 matches, 1 mismatch, and 2 gaps. And we can assign a score for each match. In this case, we assign the score +3. For mismatch, +1, and for insertion deletion, -1, to identify the optimal alignment for this scoring scheme. Please note here, those scores are just based on this scoring scheme. And the first alignment got a 15 score. The second one got a 16, and the third one got a 14. So we choose the middle one, the highest score one. And usually biologists often prefer parsimonious alignments, where the number of the postulated sequence changes is minimized. So we choose the middle one because it has the most number of matches and the least number of mismatch or gaps. But be cautious. There may be more than one optimal alignment, and even the optimal alignment may not reflect the true evolutionary history of our sequences. So there is a side note, the sequence identity and the similarity. These are the two commonly coded metrics for pairs of aligned sequences. Identity typically codes the percent of identical characters in the aligned region of two sequences. So A has to be exactly same to A. G has to be exactly same to G. And the similarity depends on the parameter used or the scoring scheme used. For example, if in my scoring system, I define the two ring purine A and G are 100% similar, the one ring pyridine C and T are 100% similar, then the sequence may be of 100% similarity, but only have 80% identity. Meanwhile, in contrast, homology is an all or nothing relationship. You cannot have a percent homology.